right, well, welcome everyone. I'm so happy that we're all here to welcome Dr. David Reiter for his talk today. He's an associate professor of radiology, orthopedics, and biomedical engineering at Emory. He focused his graduate and doctoral training in mechanical and aeronautical engineering at UC Davis. And at Emory, his research program focuses on applying engineering principles to advancing imaging diagnosis and treatment of musculoskeletal injury disease, and now also endocrinology. And with that, I welcome Dr. Reiter. Thank you.
Thank you, Lisa, and thank you guys for the invitation to come present our work to you today. The title of my talk is Imaging Endocrine Pancreas Function in Humans, a bold approach for characterizing diabetes pathophysiology. And for those of you that are non-imagers, it's a little bit of a play on the words. BOLD is an acronym in MRI for blood oxygenation level dependent imaging. And I'm going to get into a little bit of the details of that further on in my talk, but you know, it's a new area of work that we've been working to develop to try to get at better direct markers of endocrine pancreas function, which is something that's much needed. Full disclosure, as Lisa said in my introduction, I am an engineer by training. I have training also in the musculoskeletal system, and that's primarily where I've been doing the vast majority of my work through my career. And it wasn't until I met Dr. Belanke who really encouraged me to think outside of the musculoskeletal system and consider the really important problem of diabetes and try to figure out how we can apply some of our quantitative MR methods to study the disease a little bit better. So, you know, I see myself as really a bridge scientist, as an engineer that can bring technology and imaging methodologies to the study of disease. And that's really how our laboratory is sort of positioned ourselves. But today I'm really going to try to stay in my lane and talk about the things that we're doing and sort of how that relates to pathophysiology, as I understand it, in the pancreas with diabetes. And really try to stay, you know, form a bridge in any way that we can to this ongoing work with you guys, as well as anybody else who has an interest in some of the things we're doing. Just to give a quick, just brief overview of my laboratory and the work that we focus on. As I mentioned, we do quantitative magnetic resonance imaging. And for those of you unfamiliar with this approach, magnetic resonance imaging is very flexible in terms of the amount of different types of image contrast that it can produce. This is quite different than other imaging technologies. And with this, we can merge that with, you know, collecting multiple image contrasts, as this cartoon on the left shows, and with physics models to extract out quantitative information about either the underlying microstructure, physiology, biochemistry, or function of the underlying tissue. Our laboratory really does this in sort of a bidirectional fashion between clinical imaging and basic experiments that involve phantom materials, tissue explants, and numerical simulation. Because we feel like to develop these types of techniques is critical for us to really understand all different scales of how the imaging contrast interacts, or how the imaging interacts with the actual tissue itself. As I mentioned, we primarily focus on musculoskeletal research, although thanks to Dr. Belanke and more recently Dr. Steinmetz, you know, we've been migrating these technologies into into the body and studying the pancreas. But some of the examples of diseases that we've worked on are here in the bottom right. MRI has an ability to really kind of perform multi-contrast, multi-parametric imaging, quantitative imaging, and this allows us to study multiple aspects of the tissue. This is an example of how we're studying tissue structure, function, and metabolism in the tendon. These cartoons just sort of depict different types of quantitative imaging that we've been doing, merging it with spectroscopy and animal studies and human imaging. So structure function is something that we've been pursuing in the musculoskeletal system, and certainly we can extend these sorts of things to studying the pancreas as well. Microcirculation and metabolism are other properties of tissue that we've been actively developing in the context of chronic kidney disease and diabetes as well as other muscle metabolic disorders in which we use localized spectroscopy techniques like phosphorus spectroscopy, where we can study energy metabolism in skeletal muscle. So for today, so that's just a little bit about me and what our laboratory is kind of focused on the past. Today, we're sort of moving into this uncharted territory for ourselves in my lab. We're going to be talking about the work we've been developing in the pancreas for quantitative imaging of the pancreas. I'm going to cover some things on some basic elements of pancreas pathophysiology and diabetes and current and emerging markers of its assessment as it relates to this project that we've been working on. I'll explain a little bit about what kind of information medical imaging and specifically MRI can tell us about the pancreas and diabetes. I'm going to give some information on some of the emerging MRI methods out there that can be used in the study of diabetes for pancreas imaging. And then I'll describe this bold MRI approach that we've been developing and applying. Please interrupt me at any point, people in the room, if you have questions about this. It's a little more difficult online, I think, for those, but it's not a big room, so you can always unmute if you want and chat. So I don't have to describe the significance of the problem of diabetes to you guys. You guys know this way better than I do, and I'm learning along the way, but of course it's highly prevalent and costly disease, and of course this problem is not getting better, it's getting worse. We think about diabetes in terms of defective insulin secretion of beta cells, and as well as the inability of insulin-sensitive tissues to respond to insulin that is secreted. Yet we characterize diabetes and sort of diagnose diabetes based off of elevated blood glucose levels, which is very indirect and not that specific or sensitive to the underlying pathophysiology. So it's limited. Even the most advanced methods that we have that are typically used in research studies, like clamp studies, for instance, that monitor insulin secretion and glucose levels simultaneously, at high time interval over time, are still, they're still not. They involve a variety of factors such as insulin secretion, resistance, and clearance. And so, it's kind of this combined effect of things that are happening at the system level that influence those measurements. So they're not direct measurements of the beta cells themselves. So, we can all, I think, acknowledge that having a direct measure of beta cell activity and being able to quantify that both in terms of the volume of beta cells that are active and the distribution of beta cells and their relative level of activity would be extraordinarily beneficial in terms of approaching this problem of diabetes. Furthermore, the need for better markers of early onset are critical as well, as we know clinically, because there's a high proportion of adults in the U.S. that have prediabetes, and the risk of progression to diabetes is extremely high for those that already have prediabetes. So we also know that early treatment of diabetes can result in remission from diabetes, but later treatment, of course, is not quite as efficacious, and so we really want to be able to to do something at an earlier time point, as well as know how far along somebody might be in this disease progression to better tailor some kind of therapy. We also know that there's a substantial amount of heterogeneity in this disease. So even though type 2 diabetes is one type, you guys have educated me quite a bit on, just the genetic background, just differences of ethnicity in terms of the presentation of diabetes and progression of diabetes. There's different rates of progression, and so the pathophysiology is likely different in different groups. And there's different responses to treatments and there's different levels of prognosis, whether you're childhood diabetes, gestational or ketoacidosis prone diabetes. These all have quite a different appearance in terms of how rapidly you might damage the endocrine pancreas through these elevated exposures to high blood sugar. And of course, there are some really wonderful ways that the field is approaching this into better personalized treatment. This palette model here that I'm showing, I think, is a beautiful illustration of how these different pathways can be identified, perhaps, and the severity of the individual in that pathway, can be sort of categorized, and this might help improve the treatment of the condition, as well as the sort of characterization of these different subtypes of type 2 diabetes or other. So I think given this, we know that the more we can characterize directly what's happening to the endocrine pancreas, could feed into these types of characterizations quite nicely and be quite powerful, in augmenting some of these blood markers that are also quite important and have taught us quite a bit. So I'm going to just dive into the medical imaging modalities that can be used in imaging the pancreas and diabetes. These are, of course, the three main players that we see. I don't have ultrasound up here. Yeah, ultrasound can be used to image the pancreas. It's not quite as widely used, even though it's an inexpensive modality. You typically see, computed tomography, MRI, and PET, and sometimes those hybrid systems like a PET MRI or a PET CT scanner used in these contexts. CT and PET oftentimes rely upon the use of exogenous contrast agents to really provide more specific information about either metabolism or the status of different cell types within the organs of the body. That's, of course, not desirable for screening purposes. You oftentimes see that way more widely used in sort of more acute and severe types of conditions, like cancers and heart disease and things that are going to kill people much more rapidly. We tend to like to use non-invasive techniques for screening purposes and for more wider deployment and tracking. And so, really MRI, I think, is kind of the shining star with that because it does provide non-invasive measurements. We don't need to use contrast agents for the vast majority of the things we do, and it can provide a wide range of information about the pancreas. So I'm going to focus on MRI. I will talk a little bit about some of the other modalities as they pertain to strengths that have been identified in some of the key pieces of the pathophysiology that I'll cover. But it's mostly MRI in this talk. So just starting out with kind of the gross picture of what happens to the pancreas with the progression of diabetes. MRI provides beautiful image contrast of the pancreas separate from all the other internal organs as shown in this image here. You can see that the pancreas is outlined here on this T1 weighted image, and the control subject here has a much larger appearing pancreas volume than the type 1 diabetic. When that's quantified by total volume, you can see that indeed, with the progression of type 1 diabetes, the overall volume of the pancreas decreases. Now that's great. It tells you something about the whole organ, but it doesn't tell you any specific information about, what part of the pancreas, whether it's the endocrine or the exocrine, or, what is changing inside the pancreas. But nonetheless, it's of course very important. You can see this in type 2 diabetes as well, that the overall volume of the pancreas decreases. So these are good overall gross markers of severity of disease, but they're not really that great for telling us about beta cells and beta cell function. They are limited because not all diabetes are the same. And in fact, this plot on the right shows that in cystic fibrosis patients without diabetes, that the volume of their pancreas is really not different whatsoever to patients that actually have diabetes with cystic fibrosis. So, it's helpful, but it really needs to be thought of as a sort of a high level view of what's happening to the organ. Now, in a homogeneous population, like in the DIRECT study, it's been quite successful at showing, for instance, recovery of the pancreas volume with responders to therapy, as shown in this in this figure here, where you can see that at baseline, from baseline to 24 months after remission of diabetes, that the pancreas volume increases in that group and almost approaches that of the non-diabetic cohort that was studied. So again, very good marker, something that you can always obtain when you're doing an MRI, but doesn't always have like the specificity to disease. What's happening at to the endocrine pancreas during diabetes at the cellular microstructural level? Well, histological studies will show us some key pieces that I think are important and interesting to note. Here in this control slide on the top left with this H&E stain, you can see the density of cellularity, and then the counter stain here in pink is for the beta cells, the blue is the alpha cells, and the brown stain is for T cells, which you don't really see any in the control. This is an islet that's pretty uniformly shaped and has a good distribution of those two cell types. In this insulitic islet from a type 1 diabetes patient, you can see this infiltration of T cells here and this sort of an irregular shape of the islet. And so this inflammatory stage of type 1 diabetes, would be expected to be something that you might actually be able to detect more grossly with imaging, and I'll show you some examples of that in a second. But that's sort of a specific type of cellular something that's happening at the islet level. In type 2 diabetes, you don't necessarily see that T cell activity as much as you see the deposition of this amyloid that you can see that's counterstained here in this Congo red that kind of fills in the islet and kind of creates these kind of barriers between the different cells, the beta and alpha cells within the islet, as shown here on the right. And so that's quite a different type of pathology that's occurring in type 2 diabetes. So can this sort of be, somewhat characterized with imaging? So for quantitative imaging, we can do what's called T1 mapping. I'm not going to get into the physics of this, but I do want to just illustrate that this quantitative approach is sensitive to the water's interaction with the proteins and the microenvironment that the water is sort of moving in within the pancreas. So you can see the pancreas here in green, and you can see this color lookup tells you the T1 value. And these are plotted, averaged over the pancreases of these different groups, both non-diabetic, pre-diabetic, and type 2 diabetic patients. And you can see that the T1 significantly increases with the appearance of disease, reflecting likely changes that are happening at that structural level of the pancreas. Another thing that can be measured is the apparent diffusion coefficient of water within the tissue itself. So this is a technique that MRI allows us to kind of use water as a molecular probe of how tightly spaced different parts of the tissue appear. And you can see in this is kind of a weird exploded view of in 3 space of the ADC map. And in this type 1 diabetic example, you can see these yellow spots that reflect elevated levels of apparent diffusion coefficient, which would be consistent with this inflammatory phase of the type 1 diabetic, T cell infiltration. So you would expect to see sort of a swelling and tracking of fluid that comes in. And certainly these focal points are detectable using this apparent diffusion coefficient imaging. So again, another modality easy to acquire while somebody's in the magnet. You can, if this is something you suspect to be occurring in your population. Last but not least, with MRI, you can also do what's called proton density fat fraction mapping. This is an axial view showing the pancreas here with the little red region of interest drawn on it. You can see everything white in this image is fat. And so this allows us to actually quantify the fraction of fat that's present in the internal organs. And you can see from this, this box and whisker plot on the bottom, I'm sorry, the font's a little bit small, but the control group has very low concentrations of fat in the pancreas. And as you move to prediabetes and diabetes, there's quite a bit of variability, but you can see that fat infiltration increases with disease severity. And that's also something demonstrated to be reversed with weight loss and management of type 2 diabetes. Indirect marker, likely sort of a later stage of change that's occurring, not directly measuring beta cell function per se. I want to shift gears a little bit and talk about some microvascular changes that occur in the endocrine pancreas with diabetes. And, you might think, well, what do we care? I mean, microvasculature, of course, is important. But in terms of the endocrine pancreas, even though it only occupies 1% of the pancreas by volume, it demands 20% of the blood flow of the entire pancreas, which is about 5 to 10-fold that of the extern pancreas. So a lot, pretty much when you're looking at blood flow into the pancreas, it's really there kind of supporting the function of the endocrine pancreas. And we'll, of course, see the importance of that as we get a little further into the slides. But what I want to point out about microvascular changes as we've been studying this is that it's kind of an interesting story and a little bit complicated. The vast majority of studies that are out there have been performed in these animal models, which are fantastic because we can do these destructive measures. For instance, on the bottom left, this is a confocal study of, this is an actual, in situ islet of a rodent model, showing that during exposure to elevated glucose levels, that there's an increase in blood perfusion capillaries. That's what those red lines indicate in the islet. And then during levels of low blood sugar, you see that the perfusion signal is much lower in these animals, showing that vascularity reacts to blood sugar changes in the circulation. And in fact, some isolated islet studies that are really beautifully done, it show that in these rodent islets, that animals that have sort of a pre-diabetes or a diabetes phenotype tend to show more increase in blood flow in the islet when in the presence of glucose. So suggesting that their blood flow change increases even more than normoglycemic, their normoglycemic counterparts, which is kind of interesting that you would get more blood flow in a disease state. But what I want to point out is that rodents and humans are, of course, quite different in terms of the way their islets and their vasculature are arranged. And so you notice in this cartoon on the bottom right that the density of capillaries and the distribution of capillaries are more tortuous, greater density than in humans. And so since we can't do any of these complicated invasive studies in humans, we have a limited amount of information about how their vasculature is responding in the presence of elevated blood sugar or lower blood sugar. What we do know from histological studies from donors is that the density of capillaries and the appearance of the capillaries change with disease. And you see that the vasculature looks much more pathologic with these sort of thickening of vessel walls, which you see a lot in like sort of cardiovascular disease phenotypes. So, and more vasculature, which is kind of interesting, right? Some of the human studies trying to measure blood flow in normal people and diabetes and the pancreas have been also a little bit mixed. For instance, this study using arterial spin labeling, which is a pretty advanced MRI technique for measuring blood flow changes, showed this was performed. These white bars represent during a fasted state, the measurement of blood flow. The gray bars are during a hyperglycemic clamp, and the black bars are during recovery from the hyperglycemic clamp. And what they showed in healthy and diabetics is that there was really no measurable change in blood flow in the pancreas during any of those conditions, or even between healthy and disease, which is kind of interesting. But this is a very difficult measurement to make for a variety of reasons I don't have time to get into. I'm suspicious that this is just a hard measurement to make with, with MRI, and therefore the sensitivity is low in this case, and that there's maybe something going on. That's because with some of the PET studies that have been done in Europe, where they've actually used PET imaging to study, progression of diabetes, there actually is evidence that blood flow changes occur both from a fasted to a glucose-stimulated phase, as well as between healthy and disease. So in these bar charts, black is healthy and gray is type 1 diabetic. And you can see there are significant differences throughout most of at least the whole and the head, the whole pancreas and the head of the pancreas at fasted states, and then during stimulation, and then the change in blood flow is actually relatively large in healthies and relatively unchanged or small in type 1 diabetes patients. So there seems to be some kind of abnormalities in microcirculation in the pancreas. This has also been observed with PET studies, with O15 PET studies in type 2 diabetes here, where you can see even in obese non-diabetic individuals, we can see that their perfusion at a fasted state seems to be reduced compared to normal controls. So that's kind of an interesting and important piece. And the reason why I bring up microcirculation, of course, is that, everything that the beta cell is doing is relying upon the delivery to sense the glucose, as well as the delivery of oxygen to perform metabolism. And so I'm certainly not a cellular biologist, but this cartoon is just to illustrate some of the high-level things that are happening within the beta cell during glucose-stimulated insulin secretion. You've got the GLUT1 or GLUT2 transporter, depending on if you're an animal or a human, that's on the outside of the cell that brings glucose into the cell. That is then used in the TCA cycle through oxidative metabolism. It produces ATP, rises in ATP levels, then block this potassium channel and cause a depolarization of the cell, and then it triggers this voltage-gated calcium channel to open up and allow calcium into the beta cell, and then that's what prompts exocytosis of the insulin granules. So in the center of all of this is oxidative metabolism. So if we want to look sort of at this vascular metabolic coupling of glucose-stimulated insulin secretion, and we think about the islet here, and we've got a beta cell, and we've got a capillary with red blood cells, and the capillary delivers glucose to the beta cell and oxygen. That's then metabolized through oxidative phosphorylation to produce ATP, again blocking the potassium channel, opening the calcium, voltage-gated calcium channel, and then insulin release. What then also happens, though, is this ATP serves a second purpose. It's released, adenosine is leached out back to the vessel, and it's one of the most potent vasodilators there is in the cardiovascular system. So it actually helps to dilate the arterioles to allow increased blood flow, to remove the insulin and circulate it to the rest of the body, as well as bring in freshly oxygenated blood to continue that oxidative metabolism. So how might this look on a time series? So if we have, before before any sort of ingestion or exposure to glucose, our partial pressure of oxygen in that local vicinity is relatively stable, as is the islet blood flow, and there's no insulin secretion or ATP production, or it's very minimal at best. Once we ingest or expose the cell to glucose, that's delivered. Immediately you start to have oxidative metabolism occur. That starts ramping up. You get a drop in the partial pressure of oxygen locally, as well as in the vessel itself. Then you start to see a rise in blood flow, as well as insulin secretion and ATP production, as well as adenosine leaching out of the cell. So this is all really important because ultimately we want to be able to measure that activity, that cellular activity within the pancreas. And so there's a very beautiful MRI method called BOLD MRI, or blood oxygenation level dependent MRI, that was developed decades ago in the functional MRI domain. It's way more general in terms of the fact that it's really sensing the difference in the magnetic susceptibility of hemoglobin in circulation. So when hemoglobin has oxygen bound to it, it is actually a diamagnetic material, meaning that it's not going to really have a big impact on the local field of water molecules that are in its vicinity. As hemoglobin becomes deoxygenated and oxygen gets pulled off and into the cell and out of the bloodstream, it then becomes more paramagnetic. And that creates almost like little magnetic fields within the circulation, and it causes a loss of signal. So you get these signal fluctuations that can track quite quickly and that are very responsive to metabolic activity. This was first demonstrated in 1990 by Ogawa and colleagues. And like I said, this is the foundation of the boom in neuroscience of fMRI, where in an animal, they demonstrated through all kinds of different stimuli, breathing different gas mixtures, looking at different pharmacokinetic experiments. They even injected insulin to look at different changes in metabolism of the animal, anesthesia of the animal. And what they see, as you can see, the difference between A and B is that in A, you can see much more sort of striations in the brain, the sort of loss of signal in the tissue bed. And that loss of signal is from metabolic activity that's not being compensated for by an inflow of this diamagnetic oxygenated blood. And so when they change that situation, they increase blood flow through giving them a different gas mixture. You can see this sinus vein signal increases, as well as does the signal in the tissue here. So that shows this sort of bold effect. That was when it was demonstrated first. Later, a couple of years later at MGH, they demonstrated this in humans, showing that in the visual cortex, the signal intensity during this type of imaging, changes dramatically enough to be able to detect it when you shine sort of a photic stimulation into the eye. So they were having people lie in the MRI with no light, and then they were imaging their brain, and they were looking at the visual cortex light up as they were shining light in their eyes. So you can see that that's this change in metabolic activity in the brain. So this was demonstrated and it's really formed the basis of all of these amazing studies in neuroscience of brain connectivity, activity, function, etc. Right? Because it's allowed them to access previously inaccessible parts of the brain through imaging the entire volume of the brain. This can be done in a quantitative fashion. Those were just sort of what we call weighted images in that previous slide. At the early stages, they were showing this sort of contrast effect, but you can actually quantify this parameter called T2 star, which is really kind of a marker of the change of susceptibility of the blood as it becomes oxygenated and deoxygenated. So as you become more paramagnetic, T2 star decreases. So basically, as you become deoxygenated, the T2 star value goes down. As you become more oxygenated, T2 star elevates. So that's how that marker directly relates to the oxygenation status of blood in the tissue. So we thought this would be great to do in the pancreas. That was sort of our initial thoughts when Priya and I were first talking about this. Lo and behold, it had actually already been done. I noticed that this date is wrong here. I think that should be 2017, but it's definitely not 2027. But Chen et al. Had actually performed an oral glucose-stimulated bowl experiment in healthy individuals, where they had these individuals either drink water or a glucose drink while lying in the MRI, and then they would map T2 star of the pancreas dynamically after they ingested that bolus of glucose. And you can see quite nicely in healthy individuals how the T2 star drops with ingestion of glucose, and it sort of stays reduced for a while, and then in their subjects, it kind of returned back to normal. And this didn't occur when they ingested water. So that was kind of their negative control that they had for this experiment. So this was really nice. So we know that there is a bowl effect that occurs in the pancreas, or at least suggested by Chen et al. So now if we go back to our model of vascular metabolic coupling during glucose-stimulated insulin secretion, and we incorporate BOLD into that, you can see I've added this trace along the time axis here of change in T2 star signal. So you can see that this change in T2 star would be expected to decrease upon stimulation with glucose, similar to how the partial pressure of oxygen drops in the pancreas or in the beta cells. As there is an increase in blood flow that occurs, there's more oxygenated blood that would come in, as we saw that there is a hyperemia that kind of occurs during glucose stimulation in healthy individuals. And that would be thought to kind of reduce this T2 star effect or increase T2 star sort of closer to baseline, but it probably fluctuate for quite a while that metabolic activity is going on. Well, that's all great, but how does that help us diagnose whether somebody has some kind of beta cell problem, with the beta cell functional problem? I want to show some results that Lisa has been generating with one of her students that is really quite beautiful from the pancreas database data set that they've gotten access to, where they have information on sort of the metabolic changes of islets that occur during glucose stimulation, as well as the insulin secretion. So these are respirometry and perfusion analyses that were done on isolated islets from healthy individuals and individuals with type 2 diabetes. And what you can see from their initial results here, which is quite interesting, is that at low levels of glucose stimulation, the oxygen consumption in the islet of people with type 2 diabetes is elevated already at very low levels of glucose stimulation. And it's even more, it's still elevated at high levels of glucose stimulation compared with healthy. And yet their insulin secretory capacity, it's not that different at low levels of glucose, but at high levels, it's impaired. So this is interesting because it suggests that they're consuming a lot more oxygen to produce a lot less insulin. So when we go back to that model that we were sort of looking at, something that you might consider from those data to occur is that oxidative phosphorylation to produce ATP is impaired. Perhaps there's uncoupling of the mitochondria, so you're using a lot of oxygen to make not a lot of ATP, which is something that definitely happens. This mitochondrial uncoupling happens in type 2 diabetes and other cell types. But what that also would do was it would reduce not only insulin secretion, but adenosine release, and therefore your partial pressure of oxygen would remain lower. And you might expect, as I'm showing here in this black trace, a much lower T2 star signal in time compared with a healthy individual that has well-functioning beta cells. So we wanted to test this by performing this oral glucose stimulated bold experiment in healthy individuals and in individuals with type 2 diabetes. And in fact, thanks to Dr. Belanki's efforts recruiting type 2 diabetes patients for this and helping to fund this, and these students in my lab that really have like spearheaded the analysis part of this work, you can see that we've got some preliminary results showing that in control subjects, that this bold response that I'm showing you, this drop in T2 star in healthy individuals is lower than it is in patients with type 2 diabetes. You can see that, and this is statistically significant when we take things like area under the curve and average kind of like the aggregate drop in T2 star. So that's consistent with our model of sort of this coupling effect, as well as it gives us hope that we might actually be able to detect differences between different levels of beta cell function and activity. So to break this down within the diabetes subjects, we only had 4 of them, but we at least had real oral glucose tolerance measurements from these individuals. You can see that there's quite a bit of variation. This is their individual T2 star traces in time, color-coded by which subject they are. And this is their C-peptide from their oral glucose tolerance test. So you can see there's variability in their insulin secretion, and there's variability in their bold response. And in fact, just as like a fun little back of the envelope calculation, of course, we don't want to say too much with an N of 4, but when we integrated this over the 20 minutes of bold, and we integrated the C-peptide release over the first 20 minutes of C-peptide release, they correlate really strongly. It's only 4 subjects, but when you think about that, this is the average bold signal over the entire pancreas, and this is the average C-peptide, over all circulation, it's pretty remarkable that we would have a correlation like this. Now, we're kind of restricted at this point to looking at the entire pancreas and these changes in the entire pancreas, but that's quite limiting. I mean, you might ask, what's the good of that? I mean, you're measuring these sort of systemic levels of glucose and insulin in the circulation. You're really kind of watering this down. One of the reasons just that this is difficult is because when we're imaging the pancreas, of course, there's respiration and cardiac motion. This is a displacement encoded MRI of the pancreas that we collected, that my colleague, Dr. Pormateza collected, showing through the cardiac cycle, these little arrows show you the displacement of different parts of the pancreas during the R to R interval. So you can appreciate, even if we eliminate respiratory motion, you can appreciate that there's a significant amount of motion on the scale of millimeters of travel of each individual interior point of the pancreas. And that can really sort of be folded into these quantitative measurements and make it challenging. And so our work has really been moving forward. We recognize this as a limitation to really unlock this ability to see spatial and temporal variation of activation of beta cells in the pancreas, that we really want to address this. Furthermore, when we think about what a lot of these more sophisticated, high volume studies of cell counts through the length of the pancreas, this is a beautiful study that shows cell counts of delta, alpha, and beta cells in different subjects in the head, body, and tail of the pancreas, and the tremendous amount of variability there is within individuals as well as within different anatomic regions. We can all appreciate that it's critical that we have that spatial information and that we're able to track that spatial change from one time point to another through subsequent breath holds and through all the cardiac cycles that occur while we're imaging these people. We also know that these distribution of cell types change as a function of anatomic site, as shown by these studies as well. So as we're tracking disease, we would want to be able to find sort of this common space of spatial registration of this information to be able to really leverage this technology, which I think is quite powerful. And I want to just take a step back and again talk about how this has happened in the brain and in neuroscience, because I think it's a model for us for how we can actually advance our field. You know, when you think about the amount of work that's gone into developing these sophisticated pipelines that co-register, they take out warping problems or motion problems or, you know, any kind of artifacts from the image and create these pre-processed data sets that are all in a common space so they can start looking at regional variation of brain activity and connectivity of these different regions. A tremendous amount of work went into that. And this is a tissue that doesn't move around like the pancreas does in the body. So it's quite a bit easier to develop that. But it gives us this exquisite information that can really provide much finer detail about changes that occur in pathology, as you can see from this sort of graphic of brain mapping of connectivity changes that occur in pathology. So what we're trying to work towards, and this is work that we've piloted, my student Anna Horner did this analysis, that we were able to get a sequence that allowed us to do a free breathing motion corrected acquisition of the pancreas that would allow us to then have all of the pancreas volumes co-registered in time, and we could at least start to identify regional variations of activation with glucose stimulation. And I just want to point out, this is the head of the pancreas and the tail of the pancreas. You can see at baseline, the head of the pancreas has a relatively high T2 star value. After glucose ingestion, it drops. And a lot of the beta cell activity is occurring in the head of the pancreas and the tail of the pancreas. And you can see both of those regions preferentially decrease in T2 star in time after glucose ingestion. Whereas where the arteries and the arterioles come into the pancreas with fresh oxygenated blood, you can see the T2 star increases. So as we average over the entire pancreas, of course, we're washing out a lot of these effects that might become much more obvious if we're able to do this motion-corrected approach. And so this is work that we're currently pursuing, but of course looking for funding to upgrade our MRI scanners to be able to perform this type of data acquisition. And of course, I think would lead to a much better understanding of how these spatial and temporal changes occur with pathophysiology of diabetes. So with that, I see I'm a couple minutes past the target, but I'll stop by just acknowledging everybody that's involved in this work, and I appreciate all of our students and collaborators through Emory and Siemens, as well as the funding sources. And I also want to just put in a shameless plug. If anybody knows any volunteers that want to volunteer for an ongoing study of pancreas imaging that we're doing, the information is on the screen. So please take a picture of that QR code so that we can get our enrollment up. And I'll stop there for questions.
Then Kat at the hand raised.
Yeah, David, fascinating. Thank you. Great work. I mean, I'm so impressed. But one suggestion and one question. The suggestion is, Lisa and I, we are involved with another study, which involves 250 participants undergoing hyperglycemic clamps at 2 time points, like 2 to 3 years apart. So I think we are at 199 participant at the moment in terms of the first round. Whether the protocol that you've developed, whether it can be implemented in that study in a small sample, it happens to be in Hyderabad, India. That's another question, because you might get additional data. It's good to get these validations in other populations. So again, Lisa can give you more details. That's my one suggestion for you to think about. And I think they have all the equipment. It's a matter of, they even may have the resources. It's just a matter of figuring out whether we can do it. My question is really, I was thinking about ATP and phosphorylation. What about phosphorus 31 MRS? Would that have any scope here?
Yeah, the phosphorus spectroscopy is certainly been applied to some of the larger internal organs. The challenge is, for instance, it's been used in the liver, particularly in the study of diabetes. It's very specific to different types of changes in these phosphorus-containing, metabolic pathways, but its sensitivity is really low, and that's the challenge with it. Is that the nucleus itself is, even though it's in high abundance, it's just because of its magnetic properties, it's hard to get a real strong signal without studying a really large volume of tissue. So localizing that to the pancreas while the pancreas is moving and people are breathing and their heart's beating and all of that, I think could be a pretty big barrier for utilizing that technology. But I mean, it's a great suggestion and question. I just think the technology isn't quite there for being able to pull that off yet.
Yes.
Yes.
Great talk. So you mentioned, and you kind of alluded towards it to the end, about the heterogeneity between different regions of the islet or of the pancreas. And one thing I was also wondering about, just where do you expect the resolution to get? As far as, do you expect you might be able to get islet, like on an islet level, be able to look? And partially I'm asking because we know failure of islets is also very heterogeneous. So it might also be interesting if you could get higher resolution to be able to map regions of islets that still look like they're quite healthy versus other areas where it might look very dysfunctional. Yeah,
that's a great point, and it's definitely something that we've been really trying to conceptualize as we move forward. The challenge is that the spatial resolution of MRI is sort of at a limit where we'll never be able to resolve individual islets just because there is a physics limit to the technology.
Yeah.
Now the benefit though of this particular technique is that, and a lot of the techniques that we try to use with MRI is that the old effect is kind of an amplifying effect. And so what it's doing is it's creating a much bigger signal than the source of what that signal is. And so, it's really looking at that sort of that blooming effect of the susceptibility of oxy and deoxyhemoglobin traveling through the circulation. So we are going to get an averaging effect over multiple islets and over different regions. We do have multiple pixels within the head of the pancreas and the tail. I mean, we get many pixels within that volume, but each of those pixels will contain kind of a volume average of what's happening at the cellular level.
Okay.
So unfortunately, it's not ever going to be that high resolution, but I do think that it still allows us. I mean, that still hasn't impaired a lot of a lot of uses of fMRI in the brain, for instance, where it still has the same spatial limitation, but you're still able to get a lot of specificity with that time space kind of analysis.
Yeah. Are you able to superimpose other modes of imaging on top of the same pancreas?
Absolutely. In fact, that was one of the reasons I sort of led with some of the other image contrast that we can produce with MRI, because ultimately, and this is done quite a bit, where you really want to have like this composite image that gives you fat infiltration, apparent diffusion coefficient, T1, the bold effect, all kind of fused together. And that gives us an opportunity to look at, like, if there's a lot of fat infiltration in one area or in one patient group versus another, does that have any sort of noticeable effect on some of these other physiologic markers? So yes, that's definitely something that we can do and we are targeting with this.
Thank you.
Thanks.
There's a question online from Darren Olson. I'm sure I'm going to not say this correctly. The spectroscopy question seems to be an obvious enhancement with a lot of technical difficulty. But what about combining with PET markers to the more blunt but simpler?
Thank you for that question. That's a wonderful question, and it's another one we've also been discussing because, you know, we actually have a PET MRI here at Emory that's dedicated for research. And, you know, I think that there's extraordinary opportunity to look at developed PET markers. So one of the things I sort of talk negatively about is that, you know, PET marker development can be quite time-consuming, costly, and it involves a tremendous amount of resources, and it's got to go through FDA clearance. But there are already markers that can be used, like, you know, O15 water, FDG, and there's others that look at, you know, fatty acid metabolism that could be used in combination together with the bold imaging, as well as with some of the multi-contrast MRI methods, and combined together. And I do think that that's a powerful platform for studying this. It comes with additional cost, but you kind of get the MRI for free if you're going to do the PET here at Emory, because the extra time for MRI is actually occurring during the PET acquisition. So I think it's an attractive thing for us to be doing, but we just haven't gotten the momentum yet with funding yet to start pursuing that. But it's definitely a great idea, and I appreciate the question.
Venkat, go ahead.
Yeah, David, one more question, and this might be crazy, but considering that the pancreas is difficult to reach, I mean, is there space here for endoscopic MRI with a radio coil or?
So when I say difficult to reach, I think I was mainly referring to, you know, surface imaging approaches like ultrasound. MRI doesn't really have trouble reaching the pancreas per se. It's just that you have to deal with the motion of the body, of the internal organs and respiration while you're doing the imaging. That's the truth for pretty much all imaging modalities that are slower than ultrasound, for instance.
What about can it be done endoscopically? I mean, like, for example, upper GI endoscopy, get more proximus to the pancreas with a radio coil.
I've never really thought about it like that. I definitely have seen experimental situations where, you know, coils have been applied internally like that to improve sensitivity. You know, that could even possibly be a way that you could get phosphorus spectroscopy to be more direct and specific. If you had a phosphorus coil that could be put close to the pancreas endoscopically, and then you know where the measurement's coming from. I hadn't thought about that. That's a pretty creative idea. But yeah, there's no reason it couldn't be done, but I've never seen it done in this setting. Any other questions, comments?
All right, if there's no other questions, we'll conclude this seminar. Thank you so much for attending.